गुड मॉर्निंग स्टूडेंट्स. नवयुवक शिक्षण प्रसारक मंडळ चापोली, संजीवनी महाविद्यालय चापोली welcome you in e-learning platform prepared by myself, Dr. Shubhangi Prabhakarao Chavan, Assistant Professor and Head, Department of Botany. Now, Swami Ramanand Tirth Marathwada University Nanded Semester Pattern Curriculum under Choice Based Credit System CBCS for undergraduate program, Faculty of Science and Technology, subject Botany, year BSc second year, paper Plant Embryology. Now, in last lecture we have seen Microsporangium, and today we will discuss Microsporangisis process and its types of microspore trade. Now, let us start. Definition of microsporogenesis: the development of pollen grade microspores from the diploid microspore mother cell by meiosis in the microsporangia of the anther is known as microsporogenesis. The pollen grain develops from the mitotic division of the pollen mother cells. It may be of two types. First of all, successive type, and second, simultaneously types. First of all, first types, successive types. In successive types of cytokinesis, peritation wall formation takes place in two steps. First of all, first wall is formed after meiosis first, separating two haploid nuclei. Hence, a single cell is divided into two cells. This stage is known as dot. The two cells of the dot further undergo meiosis second and peritation wall. Now, next develops into cell of the produce four haploid cells. This stage is known as tetrad. First, in successive types of the division, mitotic division are followed by imbibied wall formation, resulting bilateral tetrad. This type of cytokinesis is advantageous type and is common in monocots. This is the diagrammatic representation of various stages during the successive type of cytokinesis during the microsporanginesis. Next type, simultaneous type. In this case, the nucleus of the pollen mother cells divides two times, first by meiosis first and second by meiosis second, it forming four haploid nuclei in the mass of cytoplasm of the mother cell. Now, next, the four nuclei occupy of four coronaries of the cell followed by following mother cells wall in the between the nuclei. Thus, four nuclei get arranged in tetrahedral manner along the cleavage cytoplasm. In simultaneous division, walls are formed after the formation of four nuclei. This type of cytokinesis is common is dicotyledons. Example for Mellitus alba. The diagrammatic representation of various stages during the simultaneous type of cytokinesis during the microsporanginesis. Now, next, types of microspore created. Following are of types of microspore tetrad. First of all, tetrahedral, tetrahedral tetrad. Second, isobilateral tetrad. Third, linear tetrad. 4, T-shaped tetrad. And 5, disc-set tetrad. Now, tetrahedral tetrad, in this case spherical microspore mother cell divides into 4 microspores by simultaneous cell wall formation into 2 fertile planes right angle to each other. Next, isobilateral. In this case, spherical microspores mother cells divides by 2 cells wall for successively in which plane of first cell wall formation is vertical while second cell wall formation is transverse. Thus the microspores trait process appear 2 microspores and lower 2 microspores. Next, linear state. In this case, spherical microscope mother cells divides by transverse division to forms 2 daughter cells. Each daughter cell further divides by the transverse division in the same plane such that 4 microspores remain arranged in a single row. Now, next, T-shaped trait. In this case, spherical microspores mother cells first divides by transverse division to form the upper cell and the lower cell. Then the lower daughter cell divides by the transverse division upper and upper daughter cell divides by the vertical division. Thus, 4 microspores are arranged in T-shaped forms. Now, next, disc-set trait. In this case, spherical microscope mother cells divides 2 times of produce 4 haploid nuclei which lie in 4 directions. In upper and the lower nuclei get separated from the lateral 2 nuclei by upper and lower transverse wall. When 4 microspores are arranged in this manner, then the trait is known as disc-set traits. Now, types of microspore tetrads in Architochloria. First of all, tetrahedral. Next, isobilateral. Next, linear. Next, disc-set, and last, T-shaped trait. Now, next, structure of pollen grain or microspore. The wall of the pollen grain is formed of 2 layers. The outer is known as exine and inner is known as intine. The exine is the thick outer protective covering. It is made up of 2 layers, namely an outer exine and inner indexine. The outer surface of exine is scleroplated. In the intine is the thin and uniform inner layer which is made up of pectin and cellulose. The exine is formed of sporopollenin. It is resistant to physical and biological de- degradation. Polychitin is an oily layer found on the outer side of pollen in insect pollinated species. The pollen wall at one or two loci are very thin, and this circular area are called germpore or germomature. The pollen tube comes out through the aperture during the germination. The pollen grain contain dense cytoplasm and prominent nucleus. Now, next, it consists of proteins, fats, carbohydrates, vitamins, and growth hormones. It also has mitochondria and Golgi complex. Now, in this diagram, we can clearly observe mature to called pollen grain of an angiosperms. We can see vacuoles, exine, intestine, germpore, tube cells, tube nucleus, and germinative cells. Now, next, male gametophyte structure and development of male gametophyte. First of all, development of male gametophyte. The pollen tube containing the male gametophyte is called male gametophyte. The microspores or pollen grain is the first cell of male gametophyte. After its release from the anther, it undergoes expansion, which is followed by vacuolation. As a result, the nucleus is pushed to the periphery. The pollen grain undergoes first mitotic division to form two unequal cells. The larger cells is known as vegetative cells and smaller cells is known as generative cells. The generative cells at the earlier stage is hemispherical and remains attached to the pollen wall. Later, the generative cells get detached from the wall and become spherical. The vegetative cells is bigger than the generative cell. The vacuoles of the vegetative cell disappears. Now, next, the cell organelles like plastids, ribosomes, mitochondria, RNA, contents and proteins increases. The vegetative cells is larger than the nucleus of generative cells. DNA contents is less. The generative cells is spherical and it is smaller than the vegetative cell. The cytoplasm of generative cells is highly reduced and hyaline in appearance. It lacks and receives food material and RNA. The DNA contents is very high. It contains the cell organelles like microtubules, dactozones. The pollen grains are shed from the another usually in two called stage. Further development of male gametophytes occurs after raising the stigma. The intine of the pollen grain produced through one of the germpore. It formed the delicate tubular structure called the pollen tube. It was first discovered by MEC 1823 in Proteolacus, Poland. The pollen tube grows through the style. Now, next we can see germination of pollen grain and formation of male gametophytes in an angiosperm. First of all, germ pores. We can see exine, intestine and cytoplasm. Now, next figure B, generative cell. Figure B, C, D and E are generative cell. First of all, figure B, tube or vegetative cells. Figure C, tube nucleus cellulose wall, and figure D, vacuole, and last figure E, pollen tubes, male gametophyte, tube nucleus, then cytoplasm, cytoplasmic stem, cellulose flux. The diagrammatic representation of germination of pollen grain and formation of male gametophytes in angiosperms. Next, the pollen grain may be monospherous or polysphenous. When the pollen grain protrudes a single pollen tube, as in majority of angiosperms, it is known as monosphenous. If it produces more than one pollen tube, the pollen tube is known as polysphenous. Example, malva. The pollen tube is branched in cucurbita and papaya. Now, next, the vegetative and generative cell of the pollen grain migrate into the pollen tube. As the pollen tube goes through the style, cellulose plug are formed in the pollen tube at regular distance behind or growing tip of the pollen tube. The vegetative nucleus moves first inside the pollen tubes. Now it is called the pollen nucleus. The tube nucleus is assumed to play an important role in descending the through of pollen tube. The generative cells divides mitotically to form two oval or epiphytical male gametes or sperms. The formation of two male gametes or sperms from the generative cells is known as spermatogenesis. In majority of angiosperms, the G cells divides to form two sperms just prior to the entry of the pollen tubes into the embryo sac. Now, next, the male gametes are formed by the division of G cells are haploid. The male gametes are bounded by plasma membrane. Each male gamete contains a distant nucleus which prominent nucleus and cytoplasm with the vacuole organelles. Thus, the male gametophytes produce two male gametes or sperms. During fertilization in embryo sac, one of the sperm fuse with egg to the zygote and other fuse with the secondary nucleus to form the primary endosperm nucleus. Now, the diagrammatic representation of formation of sperm form microspores. Microspores mother cells, meiosis, 4 microspores, mitosis, in each microspores, vegetative cell, generative cells and two sperms.